Very nice to see after a long time. Are you able to hear me?
Am I audible?
Yeah. Please. Yes. How are you? Voice கேக்குதா?
Yes, sir. Yes, sir.
Mute பண்ணிருக்கேன் அப்படியே. Hello. Are you able to hear me? Dr. Vishwa?
Yeah.
I
Now
think
clear.
your microphone is muted.
Good morning, sir. This is so. Good morning, Professor.
Yeah. Hello. Good morning. Good morning, sir.
Hello, good morning. How are you, sir?
Fine. Thank you. How are you, sir?
Fine, fine. Really, we are very happy to meet you.
Yeah.
Thank
It's
you.
all because of Prof. Jaffer, sir.
Yes,
He
yes,
contacted me recently,
really.
and then
We
I
are
said,
very much
okay, I
happy.
can schedule one day.
Okay, okay.
And thanks, Dr. Loganathan, for coordinating this and helping Prof. Jaffer, sir.
Of course, sir. We little 5 minutes. We'll wait for the participants to join, and then we'll proceed, sir. Okay.
Yeah, sure.
A very short while we'll wait. Otherwise, we'll immediately start. Permission கேட்டுட்டு ஆரம்பிச்சிருங்க. ஆரம்பிக்கலாம். யார் மாடரேட்டர்?
Dr. Rao, shall we start, sir?
Yes, sure, please.
Okay, sir. Bismillahir Rahmanir Rahim. Good morning to everyone present here. I extend a warm and cordial welcome to one and all for the CHEM Webinar Series 2021, organized by Postgraduate and Research Department of Chemistry, Jamal Muhammad College, Trichy. Let us seek the grace of Almighty to begin the session. May I now invite Dr. Yasin Maulana, Assistant Professor of Chemistry, to recite few verses from the Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين صدق الله العلي العظيم.
Thank you, sir. It's time to invite Dr. K. Loganathan, Organizing Secretary of CHEM Webinar Series 2021, to welcome the gathering.
Respected Secretary and Correspondent, revered Treasurer, respected Assistant Secretary, beloved Principal, Vice Principal, Additional Vice Principal, distinguished resource person, dear professors, delegates, research scholars, and my dear students, good morning. Nurturing young minds and kindle innovative thoughts to the learners is the main aim of our institution. Hence, an attempt is made to organize such a webinar series. It's my privilege to welcome management committee members, principal for this webinar series being organized by PG and Research Department of Chemistry for the benefit of our students though it is a pandemic situation. It gives me a great pleasure and honor in welcoming our today's speaker, an erudite scholar, a royal researcher, an eminent speaker, Dr. Pasupuleti Visveswara Rao, Associate Professor and Research Coordinator, Head of Unit, Non-Communicable Diseases, Research Niche Areas, Head of Internationalization Unit, Department of Biomedical Sciences and Therapeutics, Faculty of Medicine and Health Sciences, University of Malaysia, Sabah, Malaysia. Though it is a time of pandemic, Sir has accepted our invitation and spared time with us to share his views on Nano-Big miracles with small particles. I hope that you will be enlightened by his sharing. I wholeheartedly welcome you, sir. I extend my warm and cordial welcome to our HOD of Chemistry, a real source of inspiration behind these activities. Dr. M. Sini Mubarak, convener of the webinar series, Dr. A. Jafar Ahmed, Dr. M. Syed Ali Padus, coordinators, Dr. F.M. Mashood Ahmed, organizing secretary, faculty members, research scholars, delegates, and students. Once again, I welcome you all. Thank you.
Thank you very much, sir, for your nice words. May I request Dr. A. Jafar Ahmed, Coordinator of CHEM Webinar Series 2021, to introduce the resource person.
Once again a very pleasant good morning to all. It is my pleasure to introduce an eminent scholar, a good friend of not only me, but a well-wisher of Jamal Muhammad College, my dear friend, Dr. Vishwa, young and energetic researcher, a teacher, a very good human being, what I can say to introduce. He is not a simple researcher, of course. He is a researcher on par, a real teacher, worked in different universities in India and abroad, a visiting researcher to Japan, Macau, Indonesia, India, and Thailand. He has got a rich experience of 14 years of teaching and research experience in the fields of biotechnology, nanotechnology, towards biomedical applications. He is a recipient of several awards. To name a few: Young Scientist Award, Young Researcher Award, Best Faculty in Biotechnology Award, Excellence Service Award at national and international levels. He has given more than 50 keynote addresses and plenary lectures in different international conferences, including one in Jamal Muhammad College, where he was the chief guest of honor. He has written 6 books, edited, and 6 book chapters, 90 publications in peer-reviewed international journals. He is an editorial member for more than 20 journals and reviewer for more than 80 journals. Currently, he is the Faculty of Medicine and Health Sciences, University of Malaysia, Sabah, Senior Associate Professor there, also heading for the non-communicable diseases research, also heads the internationalization unit. Such a great academician, an eminent scholar, a researcher on par, has consented our invitation to deliver today's keynote address, to address not only our students, but all the researchers in and around our institution, which is organized by the PG and Research Department of Chemistry, Jamal Muhammad College. This is the 4th one of this kind in this pandemic situation. We wanted to make the situation an active, not being idle. So such an activity is being held in our department. Also, at this juncture, I would like to personally thank, and also on behalf of the management committee members, my dear head of the department, and my dear fellow colleagues and organizing secretaries, I owe my thanks to Dr. Pashupati Rao for having accepted the invitation to go over here on virtually to deliver his keynote address. Thank you very much, Dr. Rao, for being here to enlighten the young minds of not only Jamal Muhammad, also the participants from various institutions in and around. Thank you very much for the opportunity given to me to introduce such an eminent scholar, my dear friend, Dr. Vishwa. Thank you.
Thank you, sir. Thank you very much for your nice introduction. Participants are kindly asked to mute their audio and turn off their video plugin. So in case of any queries regarding the topic, you may post your queries on the chat box. It's time to invite Dr. Pashupati Rao to begin the session. Please, sir.
Yeah, thank you so much for the kind invitation. Professor Jaffer, Dr. Loganathan, and also the MC who were taking all these initiatives, and as whole Jamal Muhammad College. So I'm very happy to associate with Jamal Muhammad College to have this particular session, especially on webinar series. So I hope you all can see my slides.
Yes, sir. Please proceed, sir.
Yeah, thank you. So today's topic is Nano-Big miracles with small particles. So usually when we talk about nanoparticles, we are talking as it is very new. However, it is not really very new. So I will be discussing more details in the coming slides. And hopefully the research scholars, you can try to understand the concepts. And if you have any questions, I will be very much pleasure to answer. And once again, I thank all the management of Jamal Muhammad College and also the conveners for having me here for this international webinar series. And also, this is, as Prof. Jaffer said, this is not my first time. So this was my previous, very happy moments with Jamal Muhammad College. So you can see all eminent people on the stage from Jamal Muhammad College management. And I thank my very good friend, Dr. Sirajuddin, also on that occasion, who introduced me to Jamal Muhammad College. So let us talk about nano. Nano is actually a very minute particles or very minute things, which we can emphasize more on various types of biological actions or biological activities or any other functions. Because when we talk about nano, it seems very small. However, the applications of nano are very huge. You name whatever the fields, so the nano is existing now. And nowadays, since past 10 years or past decade, we are talking about nano a very huge manner. However, it is not very new, and it has been there existing in the nature for past few decades. So we have to learn about this particular nanoparticles, or we have to understand why we are talking about nanotechnology and what is the importance of nanotechnology, what kind of particles we consider as nanoparticles, and what kind of materials we call them as we consider them as nanomaterials. So all these things are very important when we are talking about nano. And basically, when you see any particles with a nano size, it's very small, which we cannot see with the naked eyes. So that's small the nanoparticles are. It's 1 billionth of some particles or some material. Then only you can consider it as a nanoparticles. Or then we can prepare the nanomaterials with this particular nanoparticles. And when we talk about nanoparticles or nanomaterials or nanotechnology, for me, this is not one particular technology which is just focusing on everything. It is actually a combination of different subjects or combination of different fields, such as biotechnology, nanotechnology, and information technology. When we talk about nanotechnology, nanotechnology is combination of physics, chemistry, and biology. And most of the chemical components, which means chemistry is involved, and physical components, physics is involved. Whereas we are talking about biological applications, biology is involved. However, as a biotechnologist and with expertise and experience in biotechnology or biological properties or biological activities of nanoparticles or nanomaterials, I will be more focusing on biological side of nanoparticles. However, I'll be dealing with chemistry a little bit. Mostly, I will be discussing about what are the applications, because talking chemistry to the chemistry to the chemistry experts will not give much, platform here to learn. However, I would like to give more platform to what exactly is happening towards biological side, so that whatever the particles or whatever the organic materials or organic compounds you are preparing, so those will be having lots of biological applications. That's the main focus of this talk. Here in this particular slide, you can observe various types of nano ranges or nanometers of range materials. You can observe water, which is following with 0.1 nanometers, and glucose, antibodies, viruses still under nano range, because viruses we cannot see with the naked eye. Even bacteria we cannot see with the naked eyes. However, we consider the nanoparticles between 1 to 100 nanometers in size, whereas the other nanoparticles or nano-sized materials also can be considered even sometimes in micrometer range, which we cannot even see with the nano sizes. So this is the definition of nanotechnology, is a study of the controlling of any matter at atomic or molecular scale. So whenever we talk about any particle, whenever we talk about any molecule or any atom for that matter, we will be talking in different units, and we will be discussing it in different dimensions. When we talk about nanotechnology, or when we talk about nanoparticles, or when we talk about nanomaterials, we usually discuss about the nanometer range and the properties and the shapes and how much capacity they have, how much stability they have, how they can absorb the materials in it to act in the specific targeted site, where especially on drug delivery systems. Usually when we talk about nanotechnology and drug deliveries, we talk about site directed, which means specific targets it will be approaching, and the efficacy of these nanoparticles will be to the specific targets only. And here you can observe in the second statement, what I have mentioned is the materials or the particles which are falling within the range 1 to 100 nanometers are considered as nanoparticles, which they will be having lots of platforms or lots of avenues to give more biological properties, because when you shrink the size, they will be having large surface area or large surface volume, then they can have more absorbability. When they have more absorption capacity or absorbability, then they will absorb more materials or more information within the particles. Then they will try to expose or expand or explore to that particular site. And as I told you earlier, before starting my lecture, nanotechnology is definitely not new. So we are talking about nanotechnology in a new manner or in a modern manner. That's why we are talking a bit more about nanotechnology. And now still we are exploring the possibilities, and we are exploring to get best benefit out of nanotechnology nowadays. So here in this slide, you can observe that is a physicist, American physicist, Richard Feynman. So he has actually mentioned that there is a big opportunity, there is a huge opportunity with this particular nanotechnology. However, he has mentioned it in 1959. And from then on, various types of researchers, especially chemists, physicists, and also biologists, they were slowly adapting this particular nanotechnology and trying to, imbibe into their particular fields. So I will be talking about that later. So whenever you talk about nanotechnology, since this I have mentioned in the previous slide also, so the nanotechnology, you have various types of applications. You name whatever the field existing in this particular society or in this particular world. So nanotechnology exists there, and nanotechnology applications exist there. See, some of the information I have given is medical, agriculture, and food industries, because these nanoparticles can be from chemically synthesized or physically synthesized or even biologically synthesized. So that is the reason most of the chemists are also working on nanoparticle synthesis and or nanotechnology. Even physicists are also working on nanotechnology, because earlier times only the physicists and chemists were working on nanotechnology. Later on, biologists also adapt this particular nanotechnology to delve more into the applications of nanoparticles, because until synthesis, it is only chemistry. It is definitely chemistry. Then when you delve more or going through the mechanism of actions, it is biology or any engineering aspects. In this particular slide, you can observe various types of applications, which can be information technology, food and agriculture, herbal medicine, biotechnology, energy and environment, medicine and health. Not only these particular areas, but also other various areas or other different types of expert areas are being used nanotechnology in their particular fields. So I will be more focusing on herbal medicine and health for this particular nanotechnologies. So as I have mentioned very clearly, we are really talking about old picture in a new gallery, which means, so we are definitely talking about old matter, and we are talking in a bigger way, and we are trying to explore more possibilities, because we are seeing nanotechnology or nanoparticles in a new dimension. So when you see anything in the new dimension, definitely we will have the urge to delve more and more into that, and we can try to achieve whatever we can, especially into the applications. So these are the main parts of any nanoparticle synthesis. It's processing of separation, consolidation, and deformation of materials by one atom or by one molecule. So this is how usually they define, or the processes happens in nanotechnology or nanoparticle synthesis. So these are the main processes occurs. Okay, well, as I was mentioning, there was physical synthesis, chemical synthesis, and biological synthesis. So this slide clearly explains how exactly the physical synthesis, how exactly the chemical synthesis, and how exactly the biological synthesis helps the researchers or scientists to have more beneficial nanoparticles with the wide scope or wide varieties of applications. So physical, you can see evaporation, condensation, and laser ablation. So these are the various types of methods where usually physical methods are involved to synthesize the nanoparticles. Whereas chemical synthesis, Turkish methods, and also BSS, which is burst reference synthesis, they usually concentrate to synthesize nanoparticles through these methods. However, my focus will be on the very third part here, biological synthesis. When you talk about biological synthesis, when you talk about using biological materials to synthesize nanoparticles within the nano range, so usually two options. Number one is microorganisms, number two is plant materials. And again, when you take about microorganisms, there are various types of systems, fungal systems, algal systems, which means they are using fungus as the source material, and they're also using algae as the source materials. However, my main research is mostly into plant systems. I usually take plant materials, trying to identify biological molecules, polyphenols, flavonoids, flavones, whatever the secondary metabolites available in the plant materials, and then try to synthesize the nanoparticles. So that is where my major part comes into the picture. So I usually use plant materials. Either it can be various types of explants, it can be various parts of the plants, it can be leaf, it can be root, it can be bark, okay, anything. So I usually take those particular materials and then try to synthesize silver nanoparticles or gold nanoparticles. Even with collaborations, I have worked on titanium oxide, zinc oxide nanoparticles. However, there are various types of nanoparticles are being synthesized by various types of researchers. And also some of the researchers, they have tried to delve the synthesis with bimetallic natures. So with two different metals, they try to synthesize the nanoparticles. So plant systems will be my main area where I will be talking about. So whenever we talk about fundamentals of nanoparticles, or the mechanisms or what exactly they have in their particular systems, so they usually are the fundamental building blocks of nanotechnology, these particular nanoparticles, and starting point for preparing nanostructures and medical materials and devices. Whenever the nanoparticles or nano systems we talk about, so that is the basic thing we have to discuss. We have to be discussed. The nanoparticles are the very minute things where it is a starting point to prepare anything, because nanoparticle is not everything, but nanoparticles, if you have nanoparticles, you can have your nanomaterials, because nanomaterials will be formed or will be generated or will be produced with nanoparticles. And their synthesis is an important research component, because nowadays, most of the world research scientists, they are trying to delve various types of mechanism of actions with nanoparticles or by using nanoparticles in their particular research. So these are the different types of nanoparticles, metallic nanoparticles, where usually people are focusing more. However, I have mentioned in the earlier slide that there are various types of researchers. They also work on different types of metals and different type of starting materials, different types of source materials to synthesize the nanoparticles in a broader range applications with various types of targets. Okay, here you can observe in this slide silver nanoparticles, selenium nanoparticles, gold nanoparticles, and copper nanoparticles. And these are two different approaches where you can use for your nanoparticle synthesis. Number one is bottom-up approach, and second one is top-down approach. So top-down approach is the one with the macromolecules to micromolecules to nanoparticles. So whereas the nanoparticles, that is from the top down is macro to micro, micro to nano, which means you have larger atoms or larger molecules, then you are trying to make it, cut it down into very smaller pieces. So that is called bottom-up approach, top-down approach. And bottom-up approach is where you will assemble the minute particles into nano range. So that is how two different types of nanoparticle synthesis occurs. And why it is important? Why it is definitely important for the chemists, for the physicists, and also for biologists, because of its biodegradable and non-biodegradable nature. And also the main important aspect is their very smaller size with larger absorption capacity. So that is most important whenever we talk about any materials. So they should act more, and especially when you have these particular nanoparticles with more bioavailability, that is very good for any disease treatments, for any targets as well. So that is where we usually check for the more availability, more stability of these nanoparticles. And the last point, if you observe here, provide a controlled release of medication. So since biomedical researchers or biotechnologists or bioengineering scientists, they usually try to understand what exactly these nanoparticles do within the system. And they want the nanoparticles or nano systems to be delivered in a very controlled release manner. Say, for example, cancer treatments. Say, for example, other disease treatments. So they want the drug to be released in a controlled manner. Or say, for example, nowadays, even diabetes is also one of the most burdened diseases across the world, especially India. So they try to understand what exactly it is and how nanotechnology can really help these particular diseases, either metabolic diseases, either infectious diseases, or any other diseases with nanotechnology, because they provide broad range of applications, which is number one, targeted drug delivery, and another one is controlled release of medications. So here, the medicinal plants, why medicinal plants are more important. And in India, definitely we are more depending on our own traditional systems, and we certainly use lots of vegetable foods. So that is why most of the times in India, we have lots of immunity because we are consuming lots of secondary metabolites through plants and through raw materials. whereas herbs or medicinal plants play a bigger role in human systems or human wellness due to their antioxidant capacities. Because green leafy vegetables or raw materials of the plants will certainly have higher amounts of antioxidants. However, there are some plants may have toxic substances as well. But majority of the plants, and as we already know, already standardized in India, that these plants we can use for cooking purposes, these plants or these food materials we can eat raw, and which can be acting a lot of ways in your biological systems or in your biochemical pathways or in keeping your health in a better way. So that is why most of the times I focus majoritarily on medicinal plants or herbs to synthesize the nanoparticles, because we already know the secondary metabolites, how exactly they act, and we want to check whether once we synthesize the nanoparticles, whether the same activity intact or not. And these plant-based nanoparticles are having various types of applications in many types of diseases. So that's the reason we are using medicinal plants for the research. And this is the mechanism, how exactly the nanoparticles formed or nanoparticles are being synthesized. See, here you can observe the metallic nanoparticles and also metallic nanoparticles, and they are combining with bioreducing agents. Here you can see all these hydroxyl molecules, but when we talk about biological or bioreducing agents, these are the plant materials or biological molecules. It can be carbohydrates, proteins, or other biological molecules which are present in the plants, or any kind of secondary metabolites. They usually work as bioreducing agents, and they combine with metals and they form reduced silver or gold atoms, which means once they combine together, the structures will be like this. So they will be having capping on each other. Then finally they have stabilization. Once you have reduced the metallic ions with the bioreducing agents, which have the capacity to reduce ions to atoms. So this is the growth of the nanoparticles. Then finally, this is the collision, how exactly they form the nanoparticles. Once you have the nanoparticles, then the first and foremost thing we have to check is stabilization, whether the nanoparticles are stable or not, at certain point of time. And you have to check for various intervals. This is how exactly we do, and this is the slide from my research. So far, various kinds of medicinal plants we have used and mixed with metal salts. So thereby you will be having formation of nanoparticles through the process of bioreduction and also capping. So the first and foremost thing you can confirm whether your nanoparticles are formed or not is through the physical conformation. See, here it is in light color. After formation of nanoparticles, your solution may turn into the darker color. Okay, it may be dark purple or dark blue colors. So once you can imagine that, or you can observe that dark color solutions or dark color formation, then it is confirmed that physically you can confirm that there is a formation of nanoparticle synthesis. However, it is most important that you have to confirm scientifically with various types of techniques, characterization techniques. So you can observe here is UV visible spectroscopy, XRD, FTIR, SEM or TEM, and particle size analysis, which means you can observe through UV. UV also, it's a very basic information that you have successfully synthesized the nanoparticles. Then you have to confirm whether they have the particular shapes or not. And to confirm the functional groups, you have to go with the FTIR. And SEM, TEM, yeah, if you have any doubt, you can ask me, since somebody has raised the hand, or we can discuss those things in the Q&A session. So the SEM, TEM, they usually inform about morphological and also the size of the nanoparticles. Finally, the particle size analyzer, it will give you exactly how much size your particles are, because most of the times we deal with the size of the nanoparticles when we are talking about nanomaterials or nanoparticle synthesis. And these are various types of biomedical applications. I will be discussing them very thoroughly in the coming slides. Yeah, so this is how usually, to simplify the information from the previous slide, I have given this particular, slide for all of your easiness. So the metals and biological molecules, reduction to zero valent metals, then you get your atoms. And once agglomeration of atoms to nanoparticles and the stabilization by the enveloping of biomolecules. So it's like this. And once you have this particular, biomolecules enveloped, the metallic salts or metallic atoms, then you have your nanoparticles with various size and various shapes. Okay, even some of the nanoparticles, they go beyond the sizes. However, when we observe the sizes, then we consider them as nanoparticles. Okay, let us talk about bit biology here. So oxidative stress. So this is actually a very good topic in biology and also chemistry. So this is completely, even though we are talking about biological oxidative stress, but the mechanism is completely chemistry, because it's about oxidative and reductive reactions. Because oxidative stress, in a layman term, if I would like to explain, or if I want to explain this oxidative stress in a layman language, so this is just stress in the, or this is just imbalance in the normal cells, which means once you have your reactive oxygen species or reactive nitrogen species, or in a chemistry language, we can say free radicals, if they enter into the cell, they tend to damage your cells, healthy cells. So to try to avoid that particular damaging the cells, you need to have antioxidants in your system. Your antioxidant system should be active, and your antioxidant system should be highly protective, so that whatever the free radicals enter into your body, these particular existing antioxidants in your body will fight with them. Then they try to, kill all the oxidative stress or free radicals, or they try to absorb all these free radicals with the high antioxidant capacity, so that these oxidative stress or free radicals may not be having higher capacity to damage the cells or tissues or organs. So this is how exactly it looks like. So when you have your pro-oxidants, which means these reactive oxygen species or any kind of free radicals, you have high quantity, then definitely antioxidant system will be less. Whereas you have antioxidant system in a better way, or your antioxidant system is protecting your cells or your organs or your tissues in a better way, then definitely the oxidative stress will be in a balancing manner. You can observe in this picture. Yeah, let us focus on this particular picture only. Now, this is just for your information, so you can just read it out. But if we want to understand this particular equilibrium of antioxidants and reactive oxygen species, this is how exactly it looks like. In a normal healthy individual, it usually be seen in this particular manner. And most of the times we won't be having reactive oxygen species. However, if they enter into our system, the antioxidants will try to do their best. Superoxide dismutase, catalase, GPX, GR, GST, all these antioxidant system within the body I'm talking. So within the body, they try to come into the first line defensing antioxidant is SOD, superoxide dismutase. They try to fight with reactive oxygen species. Then they try to spoil them. However, if the oxidative stress or reactive oxygen species of free radicals capacity is more than the antioxidants, they try to, rule the antioxidant status in the body, so that they try to fight with the antioxidants. If antioxidants cannot fight with their capacity, then definitely the cells get damaged, or tissues get damaged, or finally organs get damaged. If you have more oxidative stress, or more oxidants, or more reactive oxygen species, or more reactive nitrogen species, then the antioxidant system will be depleting. That is why most of the times when we have anything related to the immune system or related to any disease, we try to give more antioxidants such as vitamin C and other supplements. Yeah, this is one of the most important thing I would like to mention to all of us is hyperglycemia, which is diabetes, which is one of the major diseases in India as well. So you have all these different complications, and nanoparticle research is delving more into these particular complications, and also how to reduce the diabetes or blood glucose levels in the body. Or even before reducing the blood glucose levels, nanoparticles and nanotechnology is now working on how to sense and how to give alarm to the humans before the blood glucose levels rise. Once your blood glucose levels go beyond your limit, say for example 120 or 130, but now the WHO or the other guidelines, they are trying to reduce the levels of blood glucose levels, mg per dl. So whatever the doctor speaks, 110, 120 or 130, if it goes beyond that, the sensors will alarm you that your blood glucose levels are going up, so that your intake should be less. And even probably there might be an opportunity to have this particular nanotechnology based materials or equipments for hypoglycemic, for the people with low sugar levels in their body. And these are various different types of advantages and disadvantages, how exactly they have. But most of the time in this particular slide, it is talking about how exactly this metabolic syndrome, it is not only about diabetes, even cardiovascular diseases, any other diseases, obesity. These particular markers you can observe, oxidative stress is high, HDL, which is good cholesterol, is low, triglycerides are high, inflammation is high. So all these things goes up when you have this metabolic syndrome and when you have fluctuations or disturbances in the system. And these are all other associated diseases with high range of free radicals or oxidative stress. Yeah, nanoparticles and biological applications. So mainly we have to discuss about these things. When you have your nanoparticles, whatever the shape or size or whatever the form it is, or whatever the form they are, nano particles or nanostructures, they are highly positive with high biological activities. They are very much significant particles with various types of biological activities, including in vitro, antimicrobial, antioxidant, anti-diabetic, anti-cancer, anti-inflammatory, whatnot. So once you have confirmed that it is in vitro antioxidants, in vitro antimicrobial, in vitro anti-diabetic, so you can test on this particular mouse or rats. This is called in vivo applications. So once we inject the nanoparticles into the rat, then we test for 30 days or 45 days based on the requirement and based on the standards of the procedures or of the experimental design. Then you can find whether these particular nanoparticles are definitely playing a role in the in vivo system, because our human body is, more likely, equal to our rat systems. That is why majority of the research is happening in the rats. So even my research is working on various types of models, rat models, including wound healing, even diabetes. So even we tested in vitro diabetes. So they are very good with these kind of biological activities. However, so you can observe these particular parameters. Body weight is reduced when you have diabetes, even in rats or humans. Adipose tissue size, even you can observe these particular triglycerides, LDL and TC, total cholesterol is increased and HDL is decreased. Actually, this arrow should be here. So HDL is decreased, which is good cholesterol. And all these antioxidant systems and other inflammatory markers are being fluctuated in the diabetic system. When you give this particular plant extracts or isolated compounds or even nanoparticles, you can see the revert range of systems, how exactly they work in the in vivo systems and also in vitro systems. And this is from one of my research where we have clearly given the information about antimicrobial studies, so antimicrobial activities with various types of particles. So we have tested with Rhinacanthus nastus leaf extract. Actually, this plant is from India, and it is also available in Malaysia, Thailand, and other Southeast Asian countries. So this was the article which was published in International Journal of Animal Medicine, which is one of the premier journals in this particular field. And we have mainly focused on just antimicrobial activities those days. And this is another thing we can understand. Whenever you provide you provide nanoparticles into the systems or into in vivo system. So these are the mechanisms, how exactly they do. Mitochondria, ROS, which is a reactive oxygen species. So anything you observe here, since I am not going through the mechanisms, since this is not very important here, but the summary of this particular slide is apoptosis. Apoptosis is cell death. When you introduce your nanoparticles, it will try to kill the free radicals or reactive oxygen species so that it can, make the cell to survive more. And this is one of the article I should clearly mention about diabetes. So in this particular article, I have clearly mentioned about nano-based treatments for diabetes. And so these are various types of applications where nanotubes, artificial pancreas, nanopumps, microspheres, nanoshells. So all these things, whatever I have mentioned here in this particular article, which was published in 2014, smart tattoos. So now what I heard, even in India, these particular things are being in practice where it can monitor your blood glucose levels and it can alarm you how exactly the things happen. So this was my hypothesis in 2014 actually. So portable imaging device and tattoo using in nanotechnology, but I'm happy to know that this is now in practice. So some researchers have really done great works, and then they have finally come up with this particular smart tattoos or similar kind of devices which they can be with the skin, and then they can monitor their blood glucose levels. So these are really advantageous outbreaks in the research with the nanotechnology. And this is related to obesity. And another thing which we have done on cancerous cells. So when we introduce the nanoparticles, which we have extracted or which we have synthesized using various plant materials, then we have introduced these nanoparticles into tumor cells, cell lines. Then we have found that these particular nanoparticles are very good in, diminishing or killing the tumor cells or cancerous cell lines in vitro cultures. So the mechanism seems to be like this. They usually produce, these nanoparticles produce ROS in the system. So harming the DNA of this particular, or harming the cells of tumor cells. So that they can damage, and they can stimulate the signaling pathways. They can do apoptosis, and they can also do caspase mediated signal transduction, necrosis as well. So finally, they try to kill the tumorous cells within the normal cells. So that is how usually nanoparticles work with the target specific apoptosis or necrosis. And this is another example I would like to give in the fields of neurological disorders. So even in neurological disorders, you can observe here polymeric nanotherapeutics. So they usually go through the cells, and here also you can observe these are neural system cells and nanoparticles, nanotubes. Because why I am mentioning about polymeric nanotherapeutics, here we are not only synthesizing, as I have mentioned in one of my slides. So the nanoparticles or nanomaterials are being synthesized by using various methods, physical methods, chemical methods, and biological methods. So even polymers are also very important. Even majority of chemistry researchers are also synthesizing nanoparticles using various types of chemical compounds. And let us talk about maybe one or two slides about nanotechnology with COVID, because now we are living in the era of COVID-19 or coronaviruses. Now human beings or researchers or scientists or laymen, every day we are talking about coronaviruses. Now we are talking about phase 3. Now we are talking about Delta plus. But here, let us discuss about nanotechnology and nanoparticles or nano involving in COVID-19. Number one, COVID-19 is, COVID-19 or coronavirus is nano range. So the virus is itself in nano range, and which cannot be seen with the naked eyes. Number two is the vaccine which we are producing, or which we have now, is in the nano range, and they're also using nanoparticles in this particular drug. And finally, you can observe in this picture, this particular, even my university, some of the researchers, they have produced this nanotechnology, PPEs, personal protective equipment. So they have produced with nanosheets, and then they can protect you well. And they can even come up with the nano masks, where they can use nanomaterials to protect from the bacteria and viruses and other particles, other dust particles as well. So, okay, let us discuss this particular picture now. So number A, you can see magnetic nanoparticles, polymeric nanoparticles, quantum dots, gold nanoparticles, graphene oxide nanoparticles, liposomes, missiles, and nanotubes. And here, nanotechnology in prevention. So this is in prevention. If you use this nano coated or nano, the PPE is prepared using nanotechnology, you can prevent. And nano detections. So even to detect whether we have nanotechnology, we have virus inside, or we have produced antibodies inside after the vaccination. So you can use nanotechnology or nano detections. And finally, nanotechnology in drug delivery. So far, we have discussed in previous slides as well how nanoparticles go inside the human cells or go inside the cell lines and act, and how they generate reactive oxygen species against the free radicals. So through the drug delivery system, through even controlled release system, through specific targeted systems. So nanotechnology is really providing various types of applications, not only in the metabolic disorders, not only in infectious diseases, non-communicable diseases, even to give you more broader applications, nanotechnology is everywhere. Even nanotechnology is being used in electronics, electricals, even nanocycles you have, nano sheets you have. Even in architectures, they are using nanotechnology. Even in construction, they are using nanotechnology. Aerospace engineering, they are using nanotechnology. What not? You name anything, nanotechnology is existing. And especially cosmetics, nutraceutics, pharmaceutics, nanotechnology is existing. And nanoparticles versus COVID-19, they are usually using in diagnostics, therapeutics, and vaccines. So these are the three different types of applications using nanoparticles and COVID-19. So basically, this is about my talk for today. So hopefully we are on time. It's 10 o'clock Indian time, 12:30 Malaysian time. And these are few slides about my research in the laboratory. So basically, my research is all about this. Yeah. So nanoparticles basically play an important role in the human health with various types of ameliorating effects, which means reducing effects. Where you have high levels of free radicals, they reduce to normal level or below normal level, and they try to reduce the complications of any existing diseases. So with that, thanks to all my collaborators. Even you can see, I think this is Jamal Muhammad College. So all collaborators and also everyone, thank you.
So,
yeah, thank you so much for Jamal Muhammad College for having me here. So then we can, yeah, we can open the session for
questions.
Q&A.
Dear participants, if you have any questions, kindly post your question in chat box.
Wow, sir, I am Dr. J. Sirajuddin.
Hi, sir. Welcome.
Very, very nice presentation, sir, which is very useful to our students. Okay.
Yeah, thank you. Thank you, sir.
I thank for readily accepted our invitation. Thank you. Thank you very much, sir.
Thank you, sir.
Sir, this is Dr. Jafar. Thank you very much for the excellent presentation, lucid, clean, and crispy presentation from Professor Rao. Really, it was amazing. Let me have some clarifications from you, because I also do some good research in the field of nanosciences, nanotechnology. I'm of course going ahead with the metal oxide nanoparticles, oxides, and sometimes doping and also co-doped nanoparticles. Usually I do, as you said, green synthesis from various plant extracts, of course. But what I do is traditionally I do synthesize, characterize, and take some antibacterial, antimicrobial activities, and also all these things. Can you suggest me for any latest field where I can step on to this nanoparticle synthesis, synthesized materials?
Yeah. You mean the biological applications, or?
Yes, biological applications, or otherwise the cells, fuel cells like that, whether we can do anything.
Yeah, that's very good. If you basically, I would suggest that it is certainly depends on the researchers, interest, and also the facilities you have in your laboratory. Yes. Or you have good, if you have good collaborator, you can go with it. Second thing is, coming to your question, biofuel technology is one of the very good important areas now, very hot topic, I would say. So which has very high impact, and also which the research can be published in high impact factor journals as well.
Yeah.
And third one is when it comes to biological activities, I would say antimicrobial has come to, this is a very minimal thing.
Yes.
Where many journals are not encouraging or entertaining now.
Yeah.
Antioxidant activity and antimicrobial activity have become very meager. Maybe exhausted. Yes, definitely exhausted. Even not now. Even 10 years back, we submitted one paper. They were saying the same thing, very standard journals. But along with antimicrobial and antioxidant, if the researchers can do anti-diabetic or anti-cancer,
yes,
definitely if anti-cancer, majority of the journals are interested.
Okay. Thank you.
Yeah.
Thank you for your nice presentation, and it is very useful for the entire participants. Thank you.
Thank you, sir. It is because of you I'm here with you. So the other day you have contacted me for this particular session. Thank you for that.
Also a nice gesture.
One question from participant, Dr. P. Sugandhi asked one question, sir.
Yeah.
I have some doubts regarding the nanotoxicity of the chemically synthesized nanoparticles in humans. Is there any possible for green synthesized based nanoparticles?
Yeah, that's a very good question, Dr. Sugandhi. Actually, I have purposely removed the slide in order to have this question from the participants. And majority of my presentations, I will ask the participants to ask. If they don't ask also, I will try to ask them. This is certainly a very good question, nanotoxicity of chemically. And this is a very big debate going on among the researchers about toxicity of this particular nanoparticles. Well, when we talk about nanoparticle synthesis, we use very limited quantity or very less concentration of the metallic salts. And second thing is when we prepare the nanoparticles, once we synthesize the nanoparticles, when we are using it to the rats or even human systems, the concentration will further be reduced, which means the toxicity levels are not really significant when it goes into the in vivo system. And second thing is, coming to again the question, second explanation is, when we synthesize the nanoparticles, and if it is certainly the motivation or experimental design for the researcher to test on animals, we will test the toxicity studies or toxicity levels, IC50 levels, so that we know exactly whether these particular nanoparticles are producing any toxic effects to the rats or not. So if they are not producing any toxic effects to the rats in vivo system, and certainly we can try to modify them and we can try to fine tune them in order to, give it to the humans. Of course, if we want to give it to humans, it has to go through clinical trials and so on. However, when talking about toxicity, we will be using very less concentration of the nanoparticles because they can absorb more materials or more drugs so that they can go to the targets. So certainly the toxicity is not a big thing when we are injecting into the in vivo system, because we have already tested. The researchers usually test for antioxidants and also toxicity studies before injecting into the rats as a preliminary study. Hope I answered your question, Dr. Sugandhi.
Yes, sir. Another one question also arised from Dr. P. Sugandhi. Nanoparticles act as a blood and a brain barrier diffusing particles. Then, if we are used for medical purposes, is there any chance?
Yeah. So nanoparticles act as blood and brain barrier, okay, BBB, diffusing particles. If we are used for medical purposes, is there any chance? Yes, that is what, if you remember one of my slides, which I have given as neurodegenerative disorders applications. So certainly if the nanoparticles are being fine-tuned with all the requirements once it goes to the blood-brain barrier, or once it goes to the cardiovascular system, or once it goes to the intestinal system, it has to be protective with all the ailments, and it has to give proper protection to the cells and tissues. So then only the researchers are trying to inject or use these nanoparticles into the in vivo system or in the human cells. So certainly there is no harm in using nanoparticles with all the approvals for any types of diseases, not only for blood-brain barriers, for every other diseases as well. Because some of the advanced laboratories and also research centers are using nanoparticles in the human systems, and they are trying to use them for the betterment of the treatments. Not only nanoparticles, even nanorobots. Yeah. So, yeah, thank you.
Thank you, sir. Thank you very much for your kind responses. It's time to invite Dr. F. Masood Ahmad to propose a hearty vote of thanks.
Bismillahirrahmanirrahim. I feel immensely happy to propose vote of thanks. First and foremost, I thank the Almighty for His blessings on the conduct of today's webinar. I express my sincere gratitude to the Honorable Secretary and Correspondent, revered Treasurer, respected Management Committee members, and respected Principal of Jamal Muhammad College for their motivation and support for conducting this program. I express the deep sense of gratitude to Dr. Pasupuleti Visweswara Rao for an illuminating lecture on behalf of our college management, principal, organizing team, and on my own behalf. I sincerely thank you, sir. Thank you very much, sir. I wholeheartedly thank Dr. M. Muhammad Shihabuddin, Additional Vice Principal and Head of the Department of Chemistry, for his valuable suggestions and guidance for this CHEM Webinar Series 2021. I would like to thank the convener of CHEM Webinar Series 2021, Dr. M. Sini, and coordinators, Dr. A. Jafar Ahmed and Dr. M. Syed Ali Badusha, for their tireless effort and able guidance for conducting this program. I thank Dr. K. Loganathan, Organizing Secretary for CHEM Webinar, for his helping hands and suggestions for the smooth conduct of this webinar. I express my sincere thanks to all the participants from various institutions and our college for their enthusiastic participation. I thank Dr. Muzaffar Ghani and Mr. Kannan for the technical support provided. I would like to thank Dr. Yasin Maulana for his prayers on the beginning of the session. I thank my teachers and staff members of our department for their support and encouragement. I also thank the student friends and research scholars of our college for their active participation. Thank you, everyone.
Thank you. Dhanyavadalu, Sri Pasupuleti Visweswara Rao.
Thank you, sir. Thank you once again. Thank you, everyone. Thank you, Dr. Masood Ahmad. Yes, sir. That is correct. Dhanyavadalu.
Thank you, sir. Thank you, sir.
Yeah.
Participants are kindly requested to submit the feedback form, which is posted in the chat box. Thank you very much, sir.
Yeah. Thank you, everyone. And thank you, Jafar sir and Dr. Loganathan, for having me here, and whole Jamal Muhammad College. Hopefully, once lockdown is fully completed or fully lifted, then we can meet face to face. Thank you, everyone.
Kannan sir, thank you, sir.
Thank you, sir. Thank you, sir.
Thank you very much, sir.
Thank you.
Kanan sir, ஒரு 2 or 3 minutes wait பண்ணிக்கோங்க சார். feedback link இப்பதான்
Sure, sir. Sure, sir.
Yes, sir. After 2 or 3 minutes, we will wind up session.
Yes, sir. Yes, sir.
Post பண்ணிக்கிறேங்க.
Kannan sir.
Sir?
Kannan sir, wind up பண்ணிக்கலாம் sir.
Yes, sir. Yes, sir. Thank you, sir. Thank you.